Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal, taking you all to the next part of biomolecules. As we have seen the models of enzymes, today we are going to talk about the effect of substrate, the temperature and pH on those particular enzymes. Alright, so let's move to your first part. Why does enzyme affect according to the external condition? Now the answer for this is very simple. As the enzymes are made up of proteins. They are made up of proteins. They contains peptide bond. They contains peptide bond. And as the peptide bond we are talking about, that peptide bond is susceptible to the external condition. So the peptide bond is susceptible to external condition. Now what does this mean? To external condition. Whenever I am continuing the harsh condition in the surrounding, the peptide bond will start breaking down. Alright, so it is susceptible to the external condition means it is going to get stronger or weaker. It is going to get stronger or weaker. Whenever the strong peptide bond is formed, the enzyme activity will increase. The enzyme will become more stable, and active. While whenever I am giving harsh condition to the enzyme, that means lowering down the temperature or increasing the temperature beyond certain limit or changing my pH to extremities, in that particular condition, the enzyme three-dimensional structure disrupt, and during which the enzyme activity will be reduced down. So basically the peptide bond is basically a storage of energy. Any bond is nothing but storage of energy. And after providing that particular energy from outside, and after providing same energy from outside, the bond is going to get broken down. So bond breaks down. And that is how your peptide bond becomes susceptible to the external condition. Got my point, guys? That's why the enzyme is going to get affected based on the external conditions it is present in. So the next part for this is effect of substrate on the enzyme. If I'm talking about effect of substrate concentration, this is one of the important part which is slightly confusing, guys. Got my point? So if we are seeing this, as per the substrate concentration is increased, as per we are going on right side of the graph, the enzyme activity is going to get increased. Got it? Until one point, the enzyme activity will increase, and after which the saturation stage is going to be seen. So this one is nothing but my saturation, while in this particular case, till some point there is a linear growth of enzyme activity. So based on substrate concentration, the enzyme activity will increase linearly till one point, after which it will increase slightly, slightly, and finally, and finally it becomes saturated. Finally, it becomes saturated. Now, why the saturation has been arrived? That is completely based on the number of enzymes. If I'm considering the 100 enzymes present in a solution, and if I'm adding up all the substrate continuously till 1000, suppose there are 1000 substrate and 100 enzymes, then obviously only 100 substrates are going to get attached, while remaining 900 are still inside the container itself. So that's why the saturation, the saturation of enzyme depends on the maximum activity, the maximum activity that is nothing but your maximum product, and that means your 100% of the enzymes are should be in a working stage. So there should be 100% enzyme are occupied by substrate, and even after that, if I'm increasing the substrate concentration, that is not going to make any change in that particular enzyme activity, as all the 100% enzymes are already occupied by the substrate itself. Got my point? Next one is Michaelis Menten constant. Now, what does this mean? Whenever I'm considering this constant, this is nothing but my substrate concentration. Alright, the Michaelis Menten constant is nothing but a substrate concentration. So for the first time, you should know that this constant is nothing but it is a substrate concentration. It is a substrate concentration. Now, what does the Michaelis Menten constant is going to decide? This substrate concentration, whatever we are considered, this substrate concentration is basically the concentration of substrate present to achieve the activity of enzyme, or which is called as velocity of enzyme, to half of its maximum velocity, So if I am considering half of the maximum velocity, that is V max by 2, at that particular point, whatever the substrate concentration is present, that is nothing but Michaelis Menten constant, which is represented as Km. Got it, guys? So this Michaelis Menten constant is nothing but my substrate concentration present at the velocity of enzyme, half of its maximum velocity. So at this particular point, the velocity should be V max upon 2. Now, why half of its maximum velocity? That should be a question. It can be 1/3, it can be 1/4, it can be 1/5, but we have considered half of the maximum velocity. Why? Because we think that whenever the linear growth of graph occurs at Vmax upon 2, maximum number of enzymes are occupied by substrate. Maximum number of enzymes are occupied by substrate. And if the enzymes are occupied by substrate, their activity starts to take place. their activity starts to take place. That is, enzyme substrate complex is formed and the enzyme product complex formed, and the product is released into the vicinity of that particular cell. Got my point? So whenever I am talking about half of the maximum concentration, means there is maximum number of enzymes are occupied by the substrate. And whenever I am talking about this, that means lower the substrate concentration. Lower the substrate concentration, that means Km is also low, and that means enzyme requires less substrate for gaining for gaining maximum velocity. For gaining maximum velocity. That is nothing but your Michaelis Menten constant. So whenever I'm talking about enzymes, how fast the enzyme is going to reach to its maximum velocity, that is completely dependent on my Km. So as the Km is low, the enzyme will reach to its maximum velocity faster than other enzyme. Got my point? So Km is going to be differ for each and every enzyme. So let's write it down. Km differs for every enzyme based on its functional requirement. Based on its functional requirement. Now, what does Km do? So Km decides enzyme affinity towards substrate. So whenever I'm talking about lesser Km or the lesser value of Km, that means affinity of enzyme to substrate is very more. Got my point? So whenever I'm talking about Km is very less, that means enzyme gains maximum velocity. Enzyme gains maximum velocity at less concentration of substrate. And whenever I'm talking about this, that means the enzyme has higher affinity. The enzyme has higher affinity towards substrate. So in this particular case, the conclusion will be enzyme has higher affinity towards substrate. Got it, guys? So that is similar whenever the Km is going to be more. So whenever I'm increasing the Km, what will happen, guys? So whenever I'm increasing the Km, the enzyme gains maximum velocity after providing high concentration of substrate. That means we have to provide more substrate to that particular enzyme so that the enzyme substrate complex is going to be formed. Alright, so this is basically more substrate is required for formation of enzyme substrate complex, guys. Got it? So whenever I'm providing more substrate, then only the enzyme substrate complex is going to be formed. So in this particular case, we can conclude that this particular enzyme has lower affinity. Towards substrate. So in this particular case, the affinity of enzyme towards substrate is very low. It is completely based on the value of Km. So role of Km is basically depends on depends on value for that particular enzyme. And how does this particular value is going to be decided? It is completely decided on the activity of enzyme. So the value is calculated, or the Km is calculated based on based on activity of enzyme. Based on activity of enzyme. Got my point, guys? So let's see your next part of the enzyme. That is role of enzyme concentration in the product formation. That is product concentration. As we have seen that enzyme acts as biocatalyst, right? So enzyme is nothing but your biocatalyst. So biocatalyst, as the name suggests, it is going to speed up the reaction. It speeds up the reaction. Speeds up the reaction. As the speeding up the reaction suggests, that means the product formation will increase. Product formation will increase. Will increase. And we have seen that the maximum velocity is dependent on this product formation and number of enzyme that is nothing but my concentration of enzyme I am talking about. Got it? So this is not hard and fast rule that without enzyme the reaction should not take place, but it takes more time to cover it up. Got it? So as we have seen, on the x-axis we have taken time, while on the y-axis we have taken product concentration, and depending on the enzyme concentration added for the test, the particular product concentration is increased. So whenever I am seeing first condition, that is no enzyme, that means reaction occurs. Reaction occurs but at very slow rate. Very slow rate. Why is that so? That is completely because of because the reaction has higher activation energy. So reaction have higher activation energy. Got it? That's why the product is formed. But as the reaction rate is slow, the product concentration is also less. So as the reaction rate is slow, the concentration of product is very, very less. Got it, guys? Second case, when I am taking 1x concentration of enzyme. 1x means just n number of enzymes are basically added. So in this case, I am adding n number of enzymes. So in this particular case, if I'm talking about that after adding enzyme, I know the reaction speed is going to get higher because of lower activation energy. So in this particular case, activation energy is reduced as the enzyme is added. But because of the limited number of enzyme, as the number of enzymes are limited, in this particular case, the product formation is based on the number of enzymes. If suppose 100 number of enzymes are added, then the 100 product will be obtained if the reaction take place in 1 minute, suppose. So in this particular case, the number of enzyme is limited, then the rate of product formation and concentration is limited. But what will happen if I am adding 2x enzyme? That means 2 times that of the condition 2. whenever I am adding more enzymes in the system, whenever I'm adding more enzyme in the system, so there is addition of 2n number of enzymes. So in this particular case, you are going to see the activation energy is reduced, but as same as your condition 2. There is not as such that number of enzymes are reducing more activation energy. No, that's not the case. For each and every enzyme, the level of reduction in activation energy is same. So activation energy is reduced same as condition 2. But what will change, that is nothing but the enzyme concentration. So whenever there is number of enzymes are more, as the number of enzymes are increased, that means the product concentration is going to be increased. That means product concentration is going to be increased. But what will happen to the product concentration in limited substrate amount? In limited substrate amount. If I'm talking about the limited substrate, then even though we are adding 2x enzyme or 3x enzyme or 4x enzyme, then the time for product formation will differ. Then time for the maximum product formation will differ, but at the end, the product formation is going to be same. So in this particular case, the substrate concentration acts as limiting factor, if substrate is limited. If substrate is limited, just the time taken to reach the maximum product will differ based on the enzyme concentration. So in this particular case, we can see that the product concentration is increased, but whenever the substrate is kept limited, then based on enzyme concentration, you are going to see difference in time to achieve maximum product concentration. So whenever I want to get a maximum product concentration, then based on the concentration of enzyme, the time is going to get reduced. That means if first condition took 1 hour to get my maximum product, then after increasing the enzyme, it will reduce down to 30 minutes. So basically the time is going to get reduced in this particular case. If I'm talking about the next condition, that is effect on pH in enzyme, and if I'm considering activity of enzyme on y-axis and on x-axis there is a pH scale. So at this particular case, if we are talking about effect of pH on activity of enzyme, the pH scales ranges from 0 to 40 till 7 as my neutral point. 7 as my neutral pH. Got it? So below 7, there is going to be my acidic pH, while above 7, there is going to be your basic pH. Now as the graph is suggesting me, first of all, the enzyme activity was 0. Alright, the enzyme activity was 0. Suppose this is point A. The enzyme activity starts increasing till one point. So this particular increase, suppose we are considering this as B. At top of the point we are considering C. Then the enzyme activity starts decreasing again based on D part, and finally on E part the enzyme becomes inactive again. Alright, we have to study 5 different points on this particular graph. And why is this happening like this particular cases? If I'm talking about A condition, in this A condition, the acidic nature of surrounding is very high. Nature is very high. In this particular case, the enzyme will behave as base. The enzyme will behave as base. So during base, it will accept H plus ion. It will accept H plus ion, and it will gain net positive charge. Net positive charge on itself. Got my point? So the complete structure of an enzyme is going to get a net positive charge. So due to this net positive charge, the enzyme will suffer through repulsive forces, and there will be unfolding of the complete three-dimensional structure. Got it, guys? What is going to happen at A point? Due to the excessive acidic nature, there is going to be the enzyme acting as a base. It will accept H plus, the net positive charge will develop, and this positive interactions will be going to show repulsive forces, due to which the enzyme will unfold in its three-dimensional structure. In case of B, as we are gradually moving the pH from higher acidic to lower acidic, so acidic nature lowers down. So acidic nature lowers down. So whenever acidic nature is lowering down, that means the net positive charge on enzyme will decrease. Got my point? So whenever net positive charge on the enzyme is decreased, that means repulsive forces are also decreased. Repulsive forces are decreased, and that means enzyme gains stability. Got it? So whenever we are moving to less acidic condition, then the net positive charge is going to get reduced down, due to which repulsive forces are reduced down and the enzyme is becoming more stable. At point C, which is called as optimum pH, in which the enzyme will show Zwitter ionic form of amino acid. So there will be Zwitter ionic form. If you are not well aware about this particular ionic form, please go and check my video in case of amino acid. Alright, so optimum pH, the enzyme will show Zwitter ionic form. In this Zwitter ionic form, the net charge on enzyme is zero. That means there is balanced positive and negative charge present on the enzyme. So there will be no repulsive forces, right? So there will be repulsive forces are zero. Repulsive forces becomes zero. That means enzyme gain maximum stability. Enzyme is stable in three-dimensional structure. Alright, and due to which there will be maximum stability, and as we are more stable, we are more productive. So similarly, the enzyme is more productive. That means activity of enzyme is at its maximum, guys. Got it? It at its maximum. The C is basically the optimum pH. The pH at which the activity of enzyme is maximum, that pH is known as optimum pH. You must be remembering that from the properties of enzyme. Now case D, as the enzyme activity is reducing down, all right, the enzyme activity is reducing down, it is showing the gradual reduction. So in this case, the stability of enzyme, the stability of enzyme decreases gradually, not suddenly. Decreases gradually and not suddenly, guys. Why is that happening? Because the enzyme starts gaining negative charge, net negative charge. Now, why the negative charge? Because as the pH is increasing now, it is moving to the basic condition in the surrounding. And whenever the basic condition are going to happen, then enzyme will act as acid and it will start donating H plus ions. As the positive charge is reduced on the complete structure of enzyme, then enzyme will get net negative charge. Got it? So in this particular case, again, the repulsive force will increase and the stability will decrease. The stability will decrease. While in case of E, there is going to be net negative charge at maximum, and finally three-dimensional structure will unfolds, and due to which enzyme becomes inactive. This will be the case of point E. So this enzyme activity increases and decreases gradually in case of pH. This is one of the important point, guys, whenever we are talking about activity of enzyme with respect to pH. But whenever we are coming to the next part, that is effect of temperature on enzyme, then at that particular case, we can see that enzyme activity is increasing as the temperature is increased. The enzyme activity increases. The enzyme activity increases as temperature is increased. But till one point. But during decrease of activity, it is suddenly decreased. Like a freely falling body. Thup. So it is basically whenever the enzymes are basically showing its activity, they are gradually increasing it as per the temperature is increasing, while beyond a certain temperature, the enzyme activity will fall swiftly. So there is sudden decrease in the enzymatic activity. So this sudden decrease is shown beyond certain temperature. Got it, guys? So let's see why is this particular case happening. If we are talking about the enzymes increasing activity, that means I am making the three-dimensional structure more stable. So the stability of enzyme increases as temperature starts to increase. This is completely based on different organisms, guys. As I have told you that organisms have their normal temperature within a body. which is called as optimum temperature of those body. If we are considering humans, that is around 37 degrees Celsius. So all the enzymes are designed in such a way that they will work at its maximum activity on 37 degrees Celsius itself, and not below that and not above that particular 37 degrees Celsius. Got it? So the maximum activity is gained at that particular temperature itself. So the stability of enzyme increases as the temperature is going to be increased, but till maximum activity. But till maximum activity. And that particular temperature is called as my optimum temperature. That is called as optimum temperature, which is plotted on the x-axis of the graph. So this is my maximum activity of enzyme at its max, and if we plot this particular case on x-axis, that will come. So this point is basically nothing but your optimum temperature. This is nothing but my optimum temperature. Got it, guys? So at that optimum temperature, the activity of enzyme is at its max. So whenever I'm increasing the temperature beyond the optimum condition, whenever there is increase in temperature beyond optimum scale, at that particular case, we are providing more energy than the bond is stored. You are remembering the bond of this particular enzymes, which are nothing but peptide bonds, are storing some of the energy. And whenever we are providing more energy or equal energy to that of the peptide bonds energy, the bond will start to get break down. Right. So in this particular case, the energy provided will be so high, that the enzymes peptide bond will break down, and as the peptide bond will break down, then the complete structure of enzyme will dismantle completely. The complete structure of peptide and the complete structure of enzyme is dismantled completely. So whenever the peptide bond is broken down, the structure of enzyme dismantles completely. Got it, guys? So it is acting as a skeleton inside our human body. If our bones are broken down, then our body posture will be completely broken down. We won't have that proper body posture. Similarly, whenever the peptide bonds are broken down, then enzyme structure is going to dismantle, and that will disrupt this will disrupt the activity of enzyme suddenly. So if I'm talking about pH, there will be a gradual decrease or gradual increase, but whenever I'm talking about the enzyme activity based on temperature, that is gradual increase but sudden decrease after the temperature rises beyond the optimum scale. Got it? These were the different condition in which the enzyme is acting differently. Now, what is the significance of enzyme? As we have seen, there should not be a surprising case that one of the significance is nothing but speeding up the reaction. Got my point? So whenever the enzymes were talking about, those were acting as biocatalyst. And whenever the biocatalyst is considered, then it increases the rate of reactions in biological system. In biological system. Plus the function of enzyme. The function of enzyme is going to decide the suitability of enzyme. The suitability of enzyme. That means if enzyme has to work longer, then the Km has to be more. And if enzyme has to work for a shorter period of time, then the particular Km for this enzyme should be low. So in this case, if the enzyme has to work longer time period, then the Km for enzyme should be more, and for shorter time period, the Km is going to be less. Now, as we are talking about enzyme, the type of enzyme and its affinity is going to decide the product formation rate. So, significance of enzyme means the Km of enzyme decides the rate of product formation. The rate of product formation. So how much product is going to form in how much time? That is completely dependent on the Km of that particular enzyme. Now, what is next significance of enzyme? How does protein works? How does protein works in different condition? In different condition that can be studied. In different conditions that can be studied. Next case, if I'm talking about enzyme, then I can understand which substrate is going to show the maximum affinity. So enzyme study also helps to decide the substrate affinity. Next case is nothing but, if I'm talking about enzymes, those are completely different in some of the cases or specific for the cells. So sometimes the enzymes are specific for the cell. And whenever I'm talking about the specificity, then they can act as a cell marker proteins. So they can act as a cell marker protein. Got it, guys? So these are some of the significances of enzymes. I hope you like this video and you will show your support to this particular channel, Loops of Biology. Please do not forget to like, share and subscribe to our channels and share this among your friends. Thank you, guys. See you in the next video. Bye-bye.